Alright, in this lecture we're going to introduce some of the kind of evolution and diversity of all the animals that we're going to cover, some of the basic stuff that we need to get, so one of the more difficult sections. Although Chapter 18 covers invertebrate diversity, we're going to get kind of the basics for all of them, and we'll talk about it. So here's one of my favorite invertebrates. We already know the insects. Here's a smiley face for you, maybe. Give them 2 eyes. They actually encompass a wide variety of shapes, sizes, habitats, diets, mating habits, other characteristics, and with more than 1 million species, nearly 3/4 of all animal species, insects are exemplar for animal diversity. So when I know a bunch about kind of animal stuff and what's going on, it's because these guys exemplify so much of it. If you've got the arthropod section, you're going to cover insects, so you'll have that part. But there'll be other things as well. So here's lots of diversity. There's some cool things. This one's a wasp. This one's the blue morpho butterfly. This is one of like the coolest things to see in nature. There's another blue morpho over here. A couple of different varieties and species. Little dragonfly, some flies, cool stuff in here. Fun stuff. Okay, so we're not going to cover invertebrate diversity. I will give this one to you, and this will be a great resource as your book, is to look in Chapter 18 if you have one of the invertebrates that we'll look at. But here's what we need to start with, is we need to give this an essential question. So our essential question is just, we'll say, how do we explain animal diversity? And some of this is going to start with body plan, which is one of the things that you need to cover in your sort of presentation. So how do we explain, or how do we describe? Maybe describe is a better word. How do we describe? I'm doing this a little bit sideways, so it looks ugly. But how do we explain, how do we describe animal diversity? So animals, all of the ones that you guys have chosen or had selected for you, everything that we're covering is an animal. They're all eukaryotes, so we know they have a true kernel, true nucleus. They're all multicellular heterotrophs, and they have cells that lack cell walls. And the other thing is that animals also use ingestion, which is an eating type of, like eating food. So unlike the fungi where we just watched, which were kind of like these heterotrophs that kind of absorbed stuff from the outside, these guys are going to use ingestion, so they're processing internally. So good. Fungi absorb nutrients after digesting food outside of their bodies. These are going to do it inside the bodies instead of outside. So we can even look at the snake. A lovely, delightful picture of a giant snake eating a big, I don't know, some sort of tetrapod animal that it is devouring. That's the way they work. So what's an animal? Most adult animals are diploid or diploid. So they have 2 copies of all their chromosomes, and they reproduce sexually. Okay. So they reproduce sexually. There are all kinds of variations to these things, but for the most part, we've got diploids that use sexual reproduction. So there's egg and sperm that are produced, and they're produced via meiosis. So meiosis is the only way that we can make egg and sperm, are gametes that are haploid cells. They fuse during fertilization to form a zygote. All this stuff should be a review for you, but we're applying it to the animal. The new thing that we get is that a zygote that divides by mitosis will form a hollow ball called a blastula. So the blastula is kind of a new thing. So a blastula is just kind of a ball of cells. So the hollow ball of cells, that's a new one. But that one's kind of important. We'll look at some embryonic development a little bit later on, say like with a sea urchin, but we can look at the basic thing about what happens. So one side of the blastula, or hollow ball of cells, will fold in and become arranged to form a gastrula. Anytime you see gastra, you can think gastric. So gastric kind of is thinking about kind of some stomach, some kind of like food layers. So think about kind of your stomach, your gastric system there. That's the whole internal structure where the food's going to go through. So it establishes kind of sort of, I'm going to say stomach-like, because we're actually talking about a whole series of things, but you get the idea, like an internal tubing. I'll give you that. Okay, so we've got a couple of layers. There's an endoderm. So endo means within. So this is the derm, which is a lining. So it's a lining for the future digestive tract. Maybe we should say gastra is more like digestive. That probably makes more sense. That's all right. That's how we process it. Ecto means the outside. So this forms the outer layer, the outer lining, the derm, that will give rise to the skin and the nervous system. And then we have a mesoderm. Meso means middle. So this is sort of the middle lining that forms a middle layer, giving rise to muscles and internal organs, the stuff that's on the inside. So you're going to need to know these because some animals have all of them and then some of them don't really have all of them. So you're going to need to look at each of your species individually. Particularly, I will say the invertebrates that we'll look at, all the things basically before the non-vertebrate chordates, you're going to need to make sure that you see which do they have out of the three. Okay, so here we go. Here's an adult. Like this is a echinoderm, a little starfish. And using meiosis, it makes eggs and sperm. So separate kind of starfish will produce egg and sperm. The two will fuse together to form the zygote. The zygote will then divide by mitosis to form kind of this like 8-cell stage. It comes up, and then we've got a blastula. So you've got the whole hollow ball of cells here that grows. They will end up forming a gastrula because sort of part of it will get poked in. And so that like in-poking layer actually will lead to the entire internal digestive tract. It's a much better way to put that. So you can see the digestive tract as it becomes a larva, and then eventually it will grow through metamorphosis or sometimes just growth processes to become an adult. Okay, so it looks like a lot, but the basic here is we've got adults producing egg and sperm by meiosis, then there's a zygote to the blastula. The blastula will end up with our gastrula, kind of our internal digestive tract, and then eventually we grow to a larva or like a juvenile or a baby or a toddler or whatever, and then to an adult. Same basic principles that you know of, we're just adding a few extra stages. And to give some terminology to this, we can go with it. So after the gastrula stage, many animals develop directly into adults. Okay, and other animals like the sea star that we're looking at develop one or more larval stages. So a larva is just an immature individual that looks different from an adult animal. So if you've ever looked on my desk, there's a jar with a bug on it, or well, there's several jars with bugs in them, but one of them has a beetle, and then you see this like white grub-looking thing. The grub is the larva. It looks very, very different than the adult beetle that's also in the container. So if you're not sure what I'm talking about, you know, come ask. I'll show you that. Like, if you ever look at dragonfly larvae, they look like crazy animal things. They go by naiads, I think, is the term for their kind of larva. Also maggots to flies, those kinds of things. So a larva undergoes a major change in body form called metamorphosis. So think the butterfly, the caterpillar to a butterfly. There we go. Becomes reproductively mature after metamorphosis. It's a complete change. Clusters of master control homeotic genes. So we should have talked about homeotic genes just a little bit, and they're kind of blueprint genes that allow for this transformation of a zygote into an adult animal. Gives blueprint. Arms should go here, legs should go here, face should go here. Don't want to mess that up. You know, you don't want to mess up a homeotic gene and have a face on a butt. It would be a real problem, right? Not a good look. So the lineage that gave rise to animals is thought to have diverged from a flagellated unicont. So one of our protists from back in the day, about a billion years ago. So the oldest generally accepted fossils that have been found are about 575 to 550 million years old. Just to show you there's some diversification, but it is related to that protist that we talked about. Kind of our animal relatives there. As old relatives to animals is that protist, the unicont. So animal diversification appears to have accelerated rapidly between 535 and 525 million years ago. This we call the Cambrian period, or the Cambrian explosion. So the most celebrated source of Cambrian fossils is called the Burgess Shale, which contains this kind of cornucopia of perfectly preserved fossils. Like, they're incredible. I'm moving fast because it's not that relevant to what you're doing, but you can see in this Burgess Shale these great presses of these kind of fossils here, just how beautiful these things are pressed into the rock layer, so you can see it. This thing's only 3 centimeters long, but how detailed and intricate it is for being just 3 short centimeters. It's impressive. And we get some really cool ones. Hallucinogenia cracks me up because they've been trying to figure out for years which way is upright of this thing. It probably, maybe it walked on its little tail or something. We don't even know. But they've been trying to determine which part of hallucinogenia is upright. That's the best. But we've got some really cool, neat fossils of what that little dude or lady looks like. Alright, so the Cambrian explosion may have been caused by an increasingly complex predator-prey relationships or an increase in atmospheric oxygen. We're not entirely sure. Hard to kind of determine all that if you weren't present. You know, that time machine's not quite there yet. So much of the diversity and body form among the animal phyla is associated with variations and where and when homeotic genes are expressed within developing embryos. So where are blueprint genes at? Where are they telling us where things should be expressed? So of the 35 or so animal phyla, again, we're only doing about 14. So you guys are like, what? There's more. There are more. Actually, technically, we're only doing about like 9 phyla, but the whole point was to get a ton of diversity. So of a bunch of animal phyla, let's just disagree on the precise number, which is why we're just covering as diverse as possible. Only one phylum includes vertebrates, and those are with a backbone. So you're all within chordates if you have one with a spinal cord. That is the one type that we would talk about. So you guys would all be under phylum Chordata. Okay. If you have a vertebrate animal, amphibians, reptiles, birds, mammals, any of those are part of Chordata. So you need to give its particular class. So if you have a vertebrate, you're given its class. Okay. But there are roughly 96% of animals that are invertebrates and lack a backbone. And so that is where you go into phyla. So you will cover the phyla if you have, let's say, roundworms, flatworms, sponges, echinoderms, cnidarians, anything in there, those are your invertebrates, your inverts. Okay. All right, so this is a good place to take a break because we'll get into what you actually need to do. Everybody needs to do with their animal here in just a minute. So this is a good spot to break. You know, make sure you've got your thoughts collected, make sure you have some notes to the side, start your summary, and then come back. Okay, so this one's a really, really important one. And this is what I'll refer back to if I'm kind of reviewing your stuff and I say this is what you need to cover for body plan. So underneath one of the parts of your rubric, it says that you need to cover things for body plan. These should be the 5 things that we're looking for body plan. So I will always refer you back to this particular slide if you haven't covered these things for body plan. So what its symmetry is, presence of tissue, true tissues, number of embryonic layers, so that was that kind of endoderm, ectoderm, mesoderm business. So does it have all 3 of these? Whether it has a body cavity, so we're going to give a word for that a little bit later on, but there is a body cavity, or if it does or doesn't, and then details of embryonic development as well. So that one may be a little trickier, but at very least you've got to have these 4 things covered. Those are essential parts to getting all the points of the body plan section of your presentation, because it matters. It's going to give us some information about development. Okay, so, oh, that's right. No. You can cover development. I'm going to go back on that one. You can cover this one. This is one of my favorite things. We'll talk about it. So you will do this one. You can do all 5. So types of symmetry, either there will be none. So there is a category where there is no symmetry. So you might want to look at that. But then we'll have radial and we'll have bilateral. So radial is where it's sort of like a pizza. No matter where you cut it in half, you will always get 2 halves that are the same. So the idea is sort of like this guy, a hydra. You can cut it any kind of way and it will give you 2 halves that are equal. Okay. Bilateral is that there is only one way. We as humans have bilateral symmetry. If you go right down sort of the middle, you can see we have one side and the other. Bi means 2, lateral means sides. So it is 2-sided symmetry, as you can see here. Okay. Make these a little bit bigger on the next ones here. So there's your radial symmetry with the sea anemone, and then bilateral with the lobster. Let's go a little bit bigger here. The next one's the embryonic tissue layers. So do they have the outer layer, the ectoderm, the endoderm, the inner layer? Do we also have a mesoderm? Those are things that you're looking for. As far as embryonic development, that fifth thing you're going to need to know, is it a protostome or is it a deuterostome? So this is the fun part of it, is that the first opening to develop during gastrulation, from that whole blastula and then gastrulation where we get kind of the digestive system coming together, if the first thing that develops is a mouth, it's first mouth in Greek, so it's protostome. If the first thing that develops is the anus, then it's second mouth in Greek, so it's deuterostome. Yeah, it is how it sounds. And I will tell you this, is that humans in particular, and so this gives you a key on mammals, humans and mammals are all deuterostomes. So you, my friend, at one point in your life, were nothing but a butthole. It's one of my best jokes. No, actually, it's not even mine. It's borrowed, but it's still a good one. So there you go. Deuterostomes, look through that one. Embryonic development, so the development of a body cavity. These are things that we're going to call kind of a coelom. If it has a body cavity, it has what's called a coelom. We'll get to talk through this one a little bit more, but that's what you're looking for. And it helps protect the organs from injury. So if it has a body cavity that's fully enclosed, that kind of covered up with mesoderm, then it's a true body cavity. So you're determining, is it a true coelom or not? Again, we'll discuss it more a little bit later. All right, so biologists make hypotheses about the phylogeny of animal groups using evidence from these body plan characteristics, fossil record, and most recently we've got molecular data with DNA sequences. So that's how we can sort of make some linkages. One phylogenetic tree that's revised to reflect new information distinguishes between sponges, these eumatozoans, animals with two tissue layers and radial symmetry. So there's a little key there. If you start to see yours that has two tissue layers and radial symmetry, to the bilaterians, who are bilateral in symmetry. We'll also have protostomes versus deuterostomes, and you could go into dysosomes versus lophotrochosomes, but I would stay away from that one. These ones, though, we can discuss. That's stuff that you ought to be able to look at as you go through it. And if you need help, you can't quite figure it out, ask. That's my point. Okay, so here's sort of to show you guys, just so you can kind of get an idea, and we'll kind of quit right after this. But sponges, cnidarians, flatworms, mollusks, annelids, nematodes, arthropods, and echinoderms are all of our things that you'll have to figure out the phylum for, because these are what we call invertebrates. Okay, everything after that is from the non-vertebrate chordates on, are a part of chordata. And so these are going to be vertebrates down here. So you'll have to give the class name for that. So already sort of in this discussion, we've talked about whether you're going to need to give class or give a phylum. Now I will say a couple of these the chordates may be a couple of classes. You can ask on that one if you're not sure. But you're going to have to give me some classes. Up here in the invertebrates, we should have phyla. And the other thing is we've covered what we're looking for in body plans. So already those are some places where you can start your research to make sure it's accurate. I've done these things a while, so I've got a pretty decent idea. So this would be a really good place to start if you've got some time left over in class. Start looking those things up for your particular animal. What do you have? What are you missing? What are you not sure of? This is a good place to start research, because that's the biggest thing. Mainly content on this one. It's a big deal. Okay, so start with those. I think that's where we're at for this one. Yeah. Invertebrate diversity, I will give you the slides for those, but it matches up perfectly with your book. There's also a vertebrate section. So if you have an invert, more helpful resources, invertebrate, do the rest of chapter 18 and look for your specific animal. If you have a vertebrate, just go to chapter 19 and you'll find some basic resources there as well. So look at your animal, look at those resources. I'll put slides up online, but the same information and maybe even a bit more detail is in your book. So that would be wise for you to check that one out at home. Maybe take some pictures of it so you can bring that research to school if you don't want to bring the whole book. Totally get that. But look for your section and see if you can get some pictures of that, because that's starting research for this whole thing. Okay, and again, if you have any questions or you're not sure or you just don't understand where you're at or what you need, don't get overwhelmed. Just either email me or come to me and ask me, because we're going to have lots of time in class together. So as you work through it, come with your questions. Let's figure this stuff out together. It's not a bad section. You're only covering one type of animal. All right. So if you need it, just ask.